So let's go ahead and get started. Today is the first day covering material for exam number 5, which will cover the cardiovascular system. I'm hearing a couple of questions about the short papers, so let me discuss that with you. The short papers are due next Wednesday. The short paper, again, is a 4-paragraph review, basically, of 3 articles, right? And the first article is come from any place at all. The second and third articles must be research articles, and by research article, I mean it has an author. It has at least 3 pages in length in its original format, and it has references to other works at the end. And you'll basically write a paragraph, what you learned from each of those papers. You will then write a 4th paragraph that tells you why you chose it, kind of very personal. I chose this topic because my grandmother was dealing with this condition, and I wanted to learn more about it, or something like that. And then finally, at the end, you have 3 citations, one for each of the 3 papers, and then attach all 3 of your papers to it. So I do get a hard copy of your 3 articles, and I read many of those as I stay current with what you have found. That'll be no later than the next Wednesday, and then also make sure you upload your 4 paragraphs to SafeAssign. For that, you'll find a green checkmark file on Blackboard. Click on that, and it will instruct you to upload the body of your 4 paragraphs to that site.
For the 2 articles that are...
The 2 research articles.
We can use, like, databases, right?
Yeah, the library did it.
Like, go to the library to find them, like we can.
You can. I mean, there are beautiful library databases through our library. Right. But any place, I mean, you can get it off Google, but many articles you'll find on Google don't have an author or don't have don't have references to other works. So as long as it's coming from, as long as it meets those 3 standards, then I'm okay with it. You'll find rich resources in the library under databases. And just make sure when you're doing your searches that you're asking for full length. Check the full length box so that you only are given options for papers that you can copy or print the whole thing out. Next Wednesday will also be the deadline for the take-home exam. That is the take-home exam. You can come and go. You don't have to do it all at once, but it's basically some questions related to things we don't have time to discuss directly. So it'll talk a little bit about the digestive system, a little bit about the lymphatic system, a little bit on vision and the special senses, and a little bit on the endocrine system as well. And those are topics that we'll dive into deeper in 106. So you got those 2 things coming down. Our next exam, just as you look forward, is going to be on the 21st, I believe, Monday the 21st. That gives me 3 lectures to cover material. Today I'll primarily cover chapter 17, which deals with with blood. And then on Monday, I'll be able to discuss the heart. And then on next Wednesday, I'll be discussing blood vessels and blood pressure. And that will bring us right up to the following Monday exam, the 21st. For that, you'll already find on Mastering the 3 chapter assignments that you can do. I've already duplicated them so that you can do those homework assignments a second time for practice. Again, I'll take the higher of those 2 scores. And I've already put out the lecture quiz as well. And like last time, I've got that due at noon on Sunday so that it gives you more time afterwards to review the correct answers. So that is the next 10 days or so. And then, boy, it won't be very much long after that. We only have a couple of lectures after that where we'll be talking about the respiratory, the urinary, and the reproductive systems, and that'll make up the content for the final exam. And that'll be on Wednesday the 30th. So there's not a lot of time left, right? We've got about 3 weeks from today, and we are done with this course. Looking at the syllabus, what does it tell us? That on exam number 5, vocabulary will be 71 through 90. Okay, so I've got 3 lectures to go through 20 slides, so I'll do 7 each time. So I'll start with 71 and move forward with you. And are there any other questions or just logistical questions, any concerns for me? Okay. So your exams are graded. I have 2 students who took it later yesterday in the testing center and for whom I do not have their scores posted yet, but I will be able to make that exam available to you to start reviewing tomorrow afternoon. Okay. So let's take a look at Vocab, 70 through 78 or so today, 71 through 78. So if you have something with fill in it, lipophilic, hydrophilic, it has a love for or an affinity of. And then phobe or phobic or phobia is a fear of or an abnormal fear of something. Phragm is a partition. So diaphragm, we know means what? Dia, through, right? So a diaphragm is a partition through the midsection of the body, if you will. So we know there's that major muscle, but a diaphragm is also a word used to describe a reproductive tool, contraception. So you're creating a partition so that egg and sperm can't meet. Physis, growth. We've seen epiphysis, metaphysis, symphysis. So we've seen that physis term in the past. Physio. Here we are studying physiology, the study of nature, of how everything works, how the functions of the body work. A pilo is a hair. So the erector pilis muscle, pili muscle, is referring to that little muscle in your dermis that can pull your hair up to erect. Pino, remember pinocytosis was cellular drinking, slurping from the environment. And plasia is a growth or formation. Hyperplasia would be an excessive growth, too much growth going on, usually thought of with a tumor. Any sort of plasty is some sort of surgical repair. Rhinoplasty would be a nose job. Plantar flexion we saw was pointing with the foot. Plantar warts found on the sole of the foot. Platypus has a flat beak, if you will, so platy. There's a muscle in your neck called the platysma, which allows you to kind of stretch up your neck and stick out your lower jaw. Plegia, we've already seen hemiplegia, paraplegia, referring to paralysis. Plex, a twisted group of nerves, is a plexus. PNEA, we'll talk about when we get to the respiratory system, but you'll see that PNEA is in pneumonia or apnea, which would be a problem with breathing, sleep apnea. Pneumo, air or lungs or gases. And then pod, think going to the podiatrist for foot-related issues. There are cells called podocytes that we'll see in the urinary system that literally are little feet, cells with little foot-like structures. Poiesis, the formation of something, the making of something. Erythropoietin is a hormone that helps to control erythropoiesis, that is the formation of your red blood cells. We'll talk about that today. Poly, many. Polymer, something made with many sides or many parts. Polycythemia is a term used to describe when you have too many red blood cells. Post, after, so postpartum, after birth. And prim, primitive or first, like a primer, like a first reading book, something like that. Pre and pro both mean before in place or time. Prenatal, before birth. Prosect. If you were to take this equivalent course at GVSU, they have a dissection lab available to you with human cadavers, and those cadavers would have already been prosected for you. You would not be dissecting them yourselves, but they would have already been cut before in time to expose organs and structures for you to examine. Procto, both the anus and the rectum, we know those two words don't mean the same thing, anus and rectum, but they both have procto as a similar prefix. Pseudo, false. We've seen pseudostratified, pseudopod, all sorts of terms with pseudo in it. And psych, a pretty good TV show. Ptero, pterodactyls, right? Winged dinosaurs, so ptero is wing. Pylor, we've seen the pyloric stenosis. It literally means gatekeeper. So as you're leaving the stomach, going into the duodenum, the gatekeeper, what controls all that, is the pyloric sphincter. If a bacterium forms pus, we might say that it's pyogenic. Pyo meaning pus. If that organism causes fever, we might say pyrogenic. So look at those two terms very carefully. You may also know a pyro, right? Someone who loves fire, heat. And then finally quad, as in quadriceps femoris, referring to fourfold or something with quad in it, quadriceps. So that'll be enough for today. And this is going to go through 90, right? So we'll start with tract, through tract. So that'll be your vocab for that test on the 21st. That'll leave just the last 10 slides, 10, 11 slides for the final exam on the 30th. vocab seems to be going really well for most people, so I'm pleased with that. Every once in a while, I still get a student who just kind of, I think they honestly forget to study it and completely blow the vocabulary, but most people are doing really well with vocab. Okay, so chapter 17 from Martini is over blood. I anticipate getting through this presentation today and probably getting a little bit started on the heart, and that should put us in good shape. Now, I'm skipping over. You have in your packet, you have notes over the special senses, yes, and you have notes over the endocrine system, and those notes are going to be part of the take-home exam. So I'm not quizzing you on it. I'm not going to be presenting it on it. I'm not having you do any mastering assignments on those chapters unless you'd like to. I could create some mastering assignments for those units, but I have not. I just left you with the take-home exam to do for those chapters. So skip over a couple of chapters and quite a few pages actually. Get past the eye and the ear, get past the endocrine system, and then pick up with the blood. And it's back on page 164. So 164 in your lecture packet for today. So some of this will look awfully familiar. This week in lab, we dealt with blood and the heart and vessels. And so some of this you have heard. Doesn't necessarily mean you have mastered it yet. And I'll be adding to what we did in lab. In lab, the major take-home lesson was blood typing. And we'll add to that today with more information about the blood. So recall that blood is a connective tissue. That means that it's loosely packed cells, the white cells, the leukocytes, the erythrocytes, and the platelets all floating around in a ground substance. In this case, that ground substance is liquid, and it's called plasma. And within that plasma, there are fibers. So it sounds like the characteristics of any other connective tissue. Blood is thicker than water. It is about 1 degree warmer, 1 degree Celsius warmer than your body temperature. And as I described in lab, you can take your blood and spin it down in a small centrifuge tube in the lab, and in do so, break the blood down into its individual components. When you break the blood down into its components, a large part of it are the erythrocytes, your red blood cells. They make up around 44-45% of the average person. They'll make up an even greater percentage in men than they do in women, as we'll describe. And these are the erythrocytes. They're going to pellet toward the bottom of the tube. Now, in the very center, in the middle of the tube, sort of at the interface of the plasma and the red blood cells, will be a very, very thin middle layer. It's referred to oddly as the buffy coat. And it's in the buffy coat that one finds the white blood cells and the platelets. So when you centrifuge the blood, that's where those cells go. And very, very small percentage. And then on top of this whole thing will be the plasma. That's the liquidy portion. It's going to make up around 55% on average. And in the plasma is where one finds the hormones, the water, the nutrients, the gases. Everything that is floating around in your blood is dissolved in that plasma. So again, to see just a visual reminder of this process, in 106, you'll actually do hematocrits, not on your own blood, but we'll have some sample bloods out, and you'll actually spin them down in a centrifuge and do something like this. But normally we take blood from a vein, and I mentioned in a couple of my lab groups why we do that. Why do we normally take blood from a vein? What's the advantage of taking blood from a vein versus an artery?
Because of the pressure in it?
Veins have less pressure, right? So when you puncture into a vein, it's not going to go squirting all over the place. So veins, and we'll talk about this as we get through these next couple of chapters, veins are under less pressure. Veins are also more superficial. So they're going to be close to the outside of the body. Your arteries, which are far more pressurized, are deeper. And if you were to sever an artery, you would know it. It's going to squirt. So an artery is going to squirt at you. A vein, the blood's just going to kind of blub, 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 come out. It's going to clot up quite easily if you don't have any clotting issues. And but what color is that blood? Right? When we take blood out of a vein, we think of veins being what color blood? Blue, right? Blue blood. We see blue on all of our models for venous blood. Now, we know that blood isn't truly blue. We perceive it as being blue through our skin. In our models, we use blue, but we know that that blood is actually more of a darker red, brick brown kind of color. So after we spin the blood down in a small tube, again, we get our hematocrit tube, and the hematocrit tube gives us a hematocrit value. And the hematocrit value here is 44%. And the reason it's 44%, if you were to take a ruler and measure this, the amount of the total blood volume that is composed of the red blood cells. So if this were the total volume up here, just measure it in millimeters, and from here to here, however many millimeters that is, and you were to measure how high that packed red blood cell were, then that would represent about 44%. So hematocrit simply is the percentage of your blood made up of the red blood cells. So just to get, again, an appreciation of the numbers, so now I do want you looking at these numbers in lab. I told you not to worry about it too much. Now we will think about the numbers. So down here in the red blood cell area, in every cubic millimeter, remember a cubic millimeter is the same as a microliter, or 1/1,000,000th of a liter, and in each microliter there would be between roughly 4 to 6 million red blood cells. Okay, minuscule little cells, just so many of them it's hard to imagine. Remember, a microliter is 1/1,000,000th of a liter, so the smallest little drop of blood you can imagine. 4 to 5, 6 million red blood cells. The buffy coat is that very thin interface. There you're going to find platelets and white blood cells. We won't be identifying the white blood cells very much this semester. We'll spend a lot of time in 106 learning how to identify those cells. I will talk a little bit more about those white blood cells, though, today. By comparison, every microliter of blood would only have about 5 to 10,000 white blood cells. So in comparison, they're rare, right? You've got millions and millions of red blood cells compared with just a couple of thousand, 5 to 10,000 white blood cells. And then there would also be about 120,000 to about 300,000 platelets per microliter as well. So a lot of platelets, but they're very, very small. So they don't take up much of the volume of the blood. Then up in the plasma, right, this is where you're going to have water, some proteins that we'll talk about in a moment. You're also going to have electrolytes and nutrients, gases, waste products, hormones, you name it. Anything else that's floating through the blood is dissolved and would be found up in this area. What's important about the plasma is that the plasma is cell-free. Okay, there are no cells in the plasma section. That's purely non-cellular. All of your cells are down below in the red or the buffy coat area. So again, when you break down the blood, we see the erythrocytes. And if you take the erythrocytes plus the elements of the buffy coat, remember what was in the buffy coat? The buffy coat were the leukocytes and the platelets, right? So we take the leukocytes and the platelets and the erythrocytes collectively, we call those, or all of that, the formed elements. The formed elements. We're not going to use the word cells here, although you will hear people say blood cells all the time. But the reason it's not accurate to say cells is that platelets aren't cells. They never were. They're little fragments of cells. So we really can't call platelets true cells. And you may remember my saying in the first day or so that red blood cells don't have a nucleus. So red blood cells can't divide. They don't have a metabolism of much. So some would argue from sort of a weird perspective that red blood cells really aren't cells either because they don't meet the standard of life. Remember, we always said cells are the basic thing of living things in our body. Red blood cells themselves can't divide. So it's kind of a stretch to call red blood cells truly cells. So we call all of these things the formed elements. Now, if you're going to go down to the blood bank and give blood, you're going to give whole blood. That is, you're giving everything, right? You're giving your formed elements and you're giving your plasma. That's your whole blood. If instead you went down to give plasma, they would do what? Take everything, but then give you back your formed elements and keep your plasma. And because the plasma is 92% water, you can go down and give plasma much more often. You can only give blood about every 6 weeks because it takes that long for your body to regenerate all those cells that have been taken from you. After you give blood, you might feel a little weak. You might feel a little anemic because you've lost a significant amount of blood. You usually give about a pint, and you only have about 5 liters of blood in you. So you're giving, a fair amount of blood away when you give at the blood bank. So let's talk about the functions of blood. Blood is one of those amazing things. It is considered a tissue again, but it has many, many functions in our body. So first we'll think about transportation. As your blood is moving around your body, you are transporting via the red blood cells, you're transporting oxygen. You're also transporting some carbon dioxide in your red blood cells. As you're transporting blood around, you're also transporting all the nutrients, right? So your plasma contains the nutrients, the glucose. It's also transporting the hormones throughout your body. And it's also carrying all the waste products. And so we're transporting, right? We're moving things around the body through the blood. We also have a very important function of regulating our body temperature. So as plasma is moving around, as all this water is moving around in your blood, it distributes and absorbs much of the heat. It's the same reason why in the summertime, Muskegon is a little bit cooler, but in the wintertime a little bit warmer, because we have that big body of water, and that big body of water is holding on to the heat, right, making us a little bit warmer in the winter and a little bit cooler in the summer. And the same thing is true as the plasma moves around our body. It helps to cool and distribute the body temperature. When we're hot, right, the blood is going to come closer to the surface of our body. We're going to get red in color, and we're going to dissipate heat. So all of this is happening as a part of the blood. We're also going to have a regulation of pH levels. We'll deal with this more completely when we get to the urinary system. But what I want you to know right now is that normally your blood has a pH of 7.4. So typically your blood has a pH of 7.4. Thought that was on here. There it is, 7.4. Slightly what? Slightly basic, right? Slightly alkaline. So your blood is not perfectly 7.0 neutral. And 7.4 is very, very important for your body to maintain its homeostasis. The enzymes in your body that are working in most of your tissues work best at pH 7.4. Remember that the pH scale is logarithmic, which means that every number is a tenfold difference in concentration. So a little bit of difference in the pH value is a rather significant change in the concentration of hydrogen. So we're going to see later on that your pH of 7.4 is very, very important, and your kidneys and your respiratory system and your cardiovascular system are all going to come together as we get over the next couple of weeks, and we'll figure out how it is that the body maintains that blood pH so tightly. Very, very important that it not change very much. There's also all this fluid moving throughout your body, and so the cardiovascular system is maintaining those fluid levels. You're always exchanging your blood plasma with your interstitial fluid. So I mentioned the lymphatic system very, very quickly in lab this week. And the lymphatic system is exchanging fluid throughout your tissues. And that fluid is going by your lymph nodes and by your tonsils and by your spleen. And it's a big part of your immune system. But the lymphatic fluid, this interstitial fluid, is also exchanging with your blood plasma. So there's this constant movement from your tissues to your blood. If this gets out of whack, then you might have edema, swelling. So if too much of the fluid leaves your blood and goes into your tissues, you would complain, oh, my ankles are swollen. Or if too much of this fluid goes back into your blood, then you might have symptoms of high blood pressure. So again, these fluid levels are very, very important as we regulate fluid in the tissues, fluid in the blood vessels. So as I mentioned here, edema versus high blood pressure. And we'll deal with those two situations as well later in the course. I'll mention this now and then I'll mention it in a couple moments. Remember from lab number 2 that saw you'd suck. Remember that phrase, saw you'd suck. And if there are, and so in order to maintain the fluid levels of your blood, we must have a set concentration of solutes in our blood. And if that solute concentration changes, if the salt concentrations, if the protein concentrations change, then either too much water will be drawn into the blood or too much water will be drawn out of the blood. So that whole idea of hypo and isotonicity and all of that is something we want to be thinking about as we think about fluid levels in our body. Finally, your blood is important in protecting you. Your white blood cells, your leukocytes are critical to your immune function. They're killing off and fighting off invading viruses and invading bacteria. You have other cells that are making your antibodies. So in the blood typing of the lab, we mentioned antibodies, and those antibodies are made by some of your white blood cells, specifically your lymphocytes. And even more specifically, your B lymphocytes. So the B lymphocytes are the cells making your antibodies. And then don't forget also under the protection that your platelets are protecting you from bleeding out. So your platelets are going to be involved with blood clotting, and clearly that's a protective mechanism as well. So if you cut your finger, cut your body, your blood should clot if all is well because of the platelets. So those are all the functions of blood. There's 4 or 5 slides there, pretty straightforward, but maybe not all of them were obvious to you before we started this conversation. So let's talk about plasma. We've talked about what's in overall blood. Red cells, white cells, platelets, plasma. Let's talk about what's in the plasma. The plasma is a mixture of water and proteins. If you took the proteins out of plasma, now you're left with what's called serum. Okay, so serum. You'll hear that word. Ladies, if you go down to Macy's or a nice place and you are looking at some of the products for your face and your body, you'll hear all sorts of serums. So sera are oftentimes animal-based products where they've taken the blood of the animal and they've taken out all the proteins, and now you have serum, and you put that stuff on your face. Okay? Lovely. So, serum, right, is pretty much plasma minus proteins, minus the clotting factors. 92% of the plasma is water. 92% of the plasma is water. So it's largely water. And what percentage of your blood is plasma? What did I tell you? About 55%, right? Roughly about 55% of all of your blood is plasma, and 92% of the plasma is water. Therefore, your blood is roughly what percentage of water? Plus or minus 50%, right? So your blood is about 50% water overall. Now that water is critical to the dissolving of substances as it travels through the body. Now you know there are some proteins in there, so let me go through after water, which is the most abundant thing. There are 4 different types of proteins in your plasma. The most abundant by far is going to be albumins. Okay, this is about 7%. So 92% water, 7% protein, 6 to 8, somewhere in that range. About 7% proteins. And the most abundant protein are the albumins. I'll tell you what those are doing. And then we get to the globulins. Then there's a protein called fibrinogen. And then finally, the rest of the proteins are collectively called the regulatory proteins. So let me start off with the albumins. The albumins. Basically egg whites, right? That's what egg whites are. They're albumins. So this is the most abundant protein in your plasma as well. It's a very small protein. It's making up a large percentage. And when you hear the word albumin in this course, I want you to think about that idea that solutes suck, and that these albumins are critical to controlling the amount of water being drawn into your blood and taken out of your blood. So albumins, think fluid control, water movement. Now, it is true that albumins do other things as well. They're going to help carry ions and help carry hormones throughout the body. But largely what I want you thinking about is this idea of regulating the water, okay? Number 2, we have the globulins. Globulins, the second largest group. You know these proteins better by their other name, and those are the immunoglobulins, aka the antibodies. So the second largest group of proteins floating in your blood are antibodies. Those antibodies are produced by the B cells, as I've already mentioned, and they're part of your immune system. Okay. Antibodies are immunoglobulins. Big part of your ability to fight off disease. The third most abundant group or type of protein is a particular protein called fibrinogen. Now, fibrinogen only makes up about 4% of your protein, but this is critical to your blood clotting. Fibrinogen is going to be in your blood all the time, but when your blood vessels are damaged, so you cut yourself, there's any kind of trauma to any part of your body, there's always blood vessels in every part of your body. What's going to happen is that fibrinogen, right, this larger fibrinogen protein is going to get cleaved, cut, and as that protein gets cut, it's going to form a protein called fibrin. Okay? Now, fibrin is insoluble, whereas fibrinogen is soluble. So this fibrinogen is always floating around in your blood, right? It is soluble. It's dissolved, if you will, in your plasma. Upon damage to your blood vessels, the fibrinogen is converted into an insoluble fibrin. And that fibrin is critical to grabbing and forming the blood clot. So fibrinogen is critical for blood clotting. Immunoglobulins were important for your immune system. Albumins were important in regulating your fluid levels. The last group, kind of a garbage pail, everything left over, all the minor proteins, are just thrown into this category called the regulatory proteins. They account for a very small amount, and basically when you go down to the blood bank, or I should say the plasma center, and if you were to donate plasma, these are the proteins that they're most concerned about. They're rather rare, each of them. These would include the clotting factors and some of the very, rare, relatively rare proteins and enzymes that are found in your blood. And what they're going to do is pool your plasma with individuals from other individuals and sell this stuff off for a pretty good dollar and make a lot of money off it. That's why they can give you some money for your time when you're donating your plasma. So we got 4 groups, right? We have the albumins for water regulation, fluid regulation, the immunoglobulins for your immune system and your antibodies, the fibrinogen, which becomes fibrin when it's forming a blood clot, and then this garbage pail called regulatory proteins. Solutes. So when I say the word solute, what comes to mind? Solutes suck. So solutes would be things like what? I just told you albumin, right? Proteins can be solutes. Glucose, sodium. Remember we use sodium as an example when we were talking about tonicity in the lab. So solutes would be anything that is going to draw water toward itself. And would you agree that plasma is an extracellular fluid? Think about it for a second. Plasma, right, the watery portion of your blood, is an extracellular fluid. It's outside of the red cells. It's outside of the white cells. And so it is not inside the cells, right? It's inside your body. It's inside your blood. But it is outside of the cells. Yes? So plasma is an extracellular fluid. And that sometimes gets us a little bit. And plasma is essentially very similar to interstitial fluid. That is that lymphatic fluid that is floating between your cells. They both pretty much have the same concentration of nutrients. They both have the same concentration of waste products and sodium and things like that. The major difference between interstitial lymphatic fluid and plasma is the concentration of oxygen. How do you think oxygen gets into your tissues? When you think about oxygen, right, comes into your lungs and it's going to go into it's going to be carried by your blood throughout your body. And then every time your blood carries oxygen down into the capillaries, down into the tissues, the oxygen jumps off, doesn't it, if you will? It gets delivered to the tissues. By what process does oxygen get delivered? What process moves oxygen from your blood into your tissues? Lab number 2. Things go from an area of higher concentration to an area of lower concentration without the use of ATP? Diffusion. Okay? So oxygen jumps off from your blood into your tissues by diffusion. And as we already know, diffusion only works if there is a difference in concentration. So if there is no difference in the concentration of oxygen in your blood with that of your tissues, then there would be no gradient, and oxygen would not jump off into your tissues. Likewise, CO2 has to be different. Right? CO2 is a waste product, right? It's a waste product of your metabolism, and CO2 is going to be made in your tissues and then is going to jump a ride back to your lungs on your blood and be exhaled. And so likewise, CO2 it moves by diffusion, it must also have a concentration gradient. So the take-home lesson from this slide is this. Plasma and interstitial fluid or lymph are essentially the same thing. The fluid between your cells in your tissues, the fluid between your red blood cells in your plasma, essentially the same, except for, major difference, is the concentration in the gases. Concentration in the gases. Does that make sense? Plasma must have higher oxygen. Interstitial fluid must have a higher CO2. And that's going to drive their exchange in the capillaries. Now, this is just a table that I grabbed from a textbook, and it's nothing here that I haven't already told you. So I like tables. I like it when someone takes all the data and puts everything into one little table or one little place, but I know these are also quite small to read. So if you wanted to grab this slide, open it up, take a look at it on your computer or tablet, you would find that pretty much everything I've said in the last 7 or 8 slides is discussed here. So again, highlights in your plasma, you've got 92% water, you've got 7% proteins. The most abundant are the albumins, followed by the globulins, then the fibrinogen, and then that garbage pail called the regulatory proteins. Also here you would find your electrolytes, nutrients, gases, and other waste products. So it just kind of lists everything I've already said to you are the components of plasma. So we've talked about what's in blood, we've talked about what's in plasma. Now let's spend a few minutes diving more into the formed elements. We'll start off with the erythrocytes, the red blood cells. They are by far the most abundant. They're going to make up more than 99% of all of the formed elements. And their job, their sole job in their life is to carry oxygen and CO2. That's the job of a red blood cell. The leukocytes, they are in comparison very rare, making up less than 0.01% of all of your formed elements. Their job is to defend against pathogens. So you've got different types of white blood cells, some of which are better at fighting off parasitic worms, some that are better at fighting off viruses, some that are better at fighting off bacteria. But regardless, you have all these different types of white blood cells that are part of your immune response. And then, of course, you've got the platelets, as we've discussed. They're helping with blood clotting. They are more numerous than white blood cells, but they're very, very small. So overall, they make up about 1% in the number of your formed elements. Again, here's a table It gives you some important facts. So this is an important table for you to stare at. Most of these details are listed somewhere else in this discussion, but this is a great place to look. So let's take a look. Erythrocytes are of a very particular size, 7.5 microns, 7.5 micrometers across. When we looked at blood cells in the lab, remember blood cells have that biconcave shape, looks like a dog biscuit or dog bone, and they have that double or that two indented sides, so we call it biconcave. And they are of a very particular size, 7.5 micrometers. If you're looking under the microscope at other tissues and you're able to see a red blood cell, it's kind of like having its own little ruler in there. Red blood cells are of a very particular size. You know, you now have a standard by which to compare everything else in your field of view. What's their job? To transport oxygen and carbon dioxide. They hang around for 120 days. So once they're formed in your bone marrow, they're going to travel throughout your body, throughout your blood, for about 4 months. That's their lifespan. And in males, their number is going to be greater than in females. In the millions, though, again, per microliter of blood. Leukocytes are larger than red blood cells, about 1.5 times to 3 times larger. They're all involved with your immune response. Now, their lifespan can be anywhere from hours to years, because some of your white blood cells are the cells making antibodies. And the reason you get vaccinated is so that your body can make antibodies, and those cells that make those antibodies can survive in your blood for 10, 20, 30 years. So we have long-lived, some of these long-lived antibody-producing cells. As far as numbers, again, 5 to 10,000 per microliter. Then finally, the platelets, they're small, really small, less than 1/4 of the size of an erythrocyte. They're involved with blood clotting, we know that. They're going to float around for only 8 to 10 days. So, a little over a week, but 8 to 10 days. So they're constantly being made, constantly being broken down. And their numbers per microliter would be in the low 100,000. So about 120,000 up to 300 or so thousand. So I want you to know the relative numbers. I'm not going to necessarily know all the numbers, but know that there are millions of red blood cells versus thousands of white blood cells versus hundreds of thousands of platelets. Know their size. Know that red blood cells are 7.5. That one is a good number to know. And then know that white blood cells are bigger and that platelets are smaller. So know these things more in their relative range, and then recognize that each of these formed elements has a very particular and different function. In lab, I introduced a hematocrit. I've already shown you that you can take the blood and split it and spin it down. We already know what this word hematocrit means. We talked about that in lab. Remember, hemato or hemo means what? Blood, and crit to separate. Remember, not hepato. Hepato is liver, but hemato is blood. And I described to you in lab that males typically, on average, are going to have a higher hematocrit value. So tell me again, what's a hematocrit value? It represents what? Hematocrit is measured in percent, right? And it's the percent of your total blood that is composed of red blood cells. So the more red blood cells you have, the higher your hematocrit. Men, 42 to 56, somewhere in that range. I always think kind of plus or minus around 50%. With women, lower than that, high 30s up into the mid 40s. Kids vary, so don't worry about the kids' ranges. And then I've also mentioned in lab that your hematocrit value can be influenced by the altitude, because at higher altitudes, there's less what? Higher altitudes, there's less pressure, and the pressure is what's critical to getting the air into your lungs. So in essence, at high altitude, your body sees less oxygen. There's just as much oxygen in the atmosphere. It's still around 21% of the gases, but there's less pressure pushing it in. So the body is saying, hey, I'm not getting enough oxygen. So in response to that, the body will start making more red blood cells. So you fly out to Denver, and your body's going to say, whoa, my brain's not happy. I'm not getting enough oxygen right now. And so it'll start making more red blood cells. And that will increase your hematocrit and increase your body's ability to transport the oxygen. It takes about 3 to 5 days for that transition to occur. So it takes about 3 to 5 days from the onset to the production of red blood cells.
Does it, if you like leave the area, then does it stop its process?
It does. So then, so we'll get to this in a moment, but it's actually your kidneys that are regulating the production of your red blood cells. Doesn't make sense yet, but it will. But your kidneys, as I'll tell you again, are constantly filtering your blood. And the kidneys are a really selective organ. And if the kidneys aren't getting enough blood and the kidneys aren't getting enough oxygen, they get really pissed off. And they stop functioning. So a person who has low blood pressure or who has not enough oxygen going to their kidneys will go into kidney failure. Okay, so for that reason, that's the way I think about it, the kidneys have an alarm system built in. When the blood pressure or the blood oxygen levels start to drop to the kidney starts to send out a hormone. And that hormone, I'll introduce it now, it's coming up in a moment, I know, but that kidney hormone is called EPO, abbreviated, or erythropoietin. Now, I just had that word in your vocab because poiesis, right, means what? Poe means to produce or make. So this is a hormone that is going to cause your body to make more erythros, right, more red blood cells. And it ends in IN, telling us it's a protein. So it's a protein hormone made by your kidneys, goes where? Where do you make red blood cells? In your bone marrow. Right? So it's made by your kidneys, travels through your blood to the bone marrow, and tells your red blood cells to be, or tells your bone marrow to make more red blood cells. Now, that would happen when you fly to Denver. Right? EPO would be produced and released by your kidneys and tell your body to increase it. But once you flew back to ground level, down to sea level, your kidneys would normalize back, you'd stop making the hormone, and your hematocrit would return. Okay? Now, I know that the hematocrit can increase in 3 to 5 days, but once those red blood cells are made, they're going to be there for 120 days. So I think it's going to take longer for the reversal process, right? So you can fly out to Denver, and in 3 to 5 days, you're going to feel better. You're going to feel more energetic, your blood levels will have normalized, your hematocrit will have increased. When you leave Denver and you come back to sea level, then you're going to have that increased oxygen levels, right, for a while. And those red blood cells are made and won't be necessarily killed off early. They'll simply slowly not be upregulated as much. So a couple more things about red blood cells. They don't have, as I've already mentioned, they don't have a nucleus. They don't have DNA, which means that they can't divide, right? They don't have the ability to do transcription and translation. They can't undergo mitosis. And so really their job is to carry oxygen. And their job is to carry CO2. And what a beautiful thing. Because remember this equation? What's that equation? C6H12O6, glucose plus oxygen, is used to produce CO2, water, and energy, ATP. If red blood cells had a metabolism, if red blood cells were doing cellular respiration, what would the red cells be doing? Gobbling up the very oxygen that they're carrying around. So this is a beautiful system in that you have these specialized cells that are carrying oxygen and themselves aren't using up the oxygen, right? So it's a super efficient system. If red blood cells had a nucleus and they had a metabolism, then they would be stealing the very oxygen that they're carrying. It's almost like, imagine an oil tanker, right? A truck going down the highway, and it's got however hundreds of gallons of fuel in the back, right, that it's transporting. And if that truck was running on the very fuel that was in the tank, right? So by the time you got from New York to California, you didn't have a whole lot left in the tank. So it's the same kind of idea. Red blood cells are carrying that oxygen very, very efficiently because they're not themselves using the oxygen that they're carrying. It's beautiful. Okay? We already know that they're biconcave in their shape. We've already described that double indentation. And there's a strange word here called rouleau. And that is that when red blood cells are going through small blood vessels, think capillaries, when the red blood cells are going through the capillaries, my fault. So think capillaries here. They can go through in basically a single file line. How big did I tell you red blood cells are? From edge to edge, they are 7.4 or 7.5 microns. Some of your small capillaries are only 8 to 10, okay, microns. So a small blood vessel, I mean, it's a rather claustrophobic squeeze, if you will, for red blood cells to go down and through your smallest vessels. And so for that reason, the red cells will kind of line up in a single file, and that's called rouleau. Now, again, these cells can't divide, and therefore they can't repair themselves, and they're being constantly broken down by your spleen and by the liver. 120 days is the average lifespan of a red blood cell. So, you know, I always think it's about 100 days. Therefore, roughly about 1% of all of your red blood cells are being destroyed every day and are having to be replaced. Back when we were talking about mutations, I think I used the word that every second we're making 25 million new cells in our body. All right, it's just astronomical. We're making 25 million new cells every second. A large number of those are red blood cells. Right? We're losing, I mean, just think about it, millions and millions of cells in every drop of blood. And so we're losing 1% or so every day, and we're having to replace them every day. So there's that shape of the red blood cells. Again, 7.5 microns. And then here it's giving you just a little example of rouleau. And you see two cells sort of lined up in a single file. It's not the best. There's another sort of one cell lined up right along the next cell. Now, the major protein in red blood cells is hemoglobin. When must red blood cells make hemoglobin? I just told you they don't have DNA. If they don't have DNA, then they can't do transcription, and they can't translate and make proteins. So that means in the maturation process, as red blood cells are being made, they must be awfully busy making this protein. So they make this protein as they're being formed, right? And then as the red blood cell matures, it can no longer make any more proteins. Every one of your red blood cells has about 280 million molecules of hemoglobin. This protein is a pigment. That means it's colorful. And it shines a bright red when it has 4 oxygens attached to it. So the maximum number of oxygens that a hemoglobin molecule can carry is 4. And when, again, all 4 are bound, then hemoglobin is bright red. And when there is less oxygen, or no oxygen, right, but less than all 4, it starts shining less than bright red. And we perceive that, again, as being a blue color. Now, I don't want you to get this word confused. Hemoglobin, right, globin, not globulin. What was globulins? The immunoglobulins were the second most abundant group of proteins in your plasma. They were your antibodies. Okay, so the globulins are the antibodies. These are globins, hemoglobin. And hemoglobin, each molecule of hemoglobin is actually a large protein made up of 4 smaller subunits, and they're not named very creatively. There are 2 alpha and 2 betas. And I'll show you a cartoon in a moment. So you have 4 little globin chains. In the center of each of these globins is a heme group. So now you see why it's called hemoglobin. You've got 4 globins with each of the globins having a heme group. Now this heme group is a non-protein unit, and it has in the very center of it an iron ion. Fe is iron. And it's to that iron that the oxygen binds. Okay, so we see iron right in the very center of the heme, and it's to the iron that the oxygen binds. So this is the connection now between iron and your oxygenation levels. If someone says they're anemic, it suggests to us that they don't have enough oxygen carrying capacity. But you'll also hear someone who's anemic say that they're low in iron. Right? So if you're low in iron, you can't have as much hemoglobin, and you can't therefore bind as much oxygen. So you'll hear the word anemic to describe both a person who's low in iron and a person who's low in their number of red blood cells and oxygen capacity to carry that oxygen around the body. So let's take a look at this, a picture of this, if you will. So hemoglobin, again, is a larger molecule made up of 4 smaller globins. In the center of them, there are iron ions that bind the oxygen. The oxygen only binds weakly, W-E-A-K. And this is important because imagine you are a red blood cell. And in your red blood cell, you've got all these hemoglobin molecules, and on each of these hemoglobin molecules, you've got oxygens attached. And your job is to transport this oxygen throughout the body. And as you're transporting the oxygen toward the capillaries, toward the tissues, that oxygen has to be able to, quote, jump off quite easily. If the hemoglobin and the oxygen were binding tightly, grabbing onto each other with great strength or great attraction, then the oxygen would not be easily able to jump off when it gets to the tissues. So the affinity, the connection between the heme and the oxygen and the iron is rather weak, so that the oxygen can easily dissociate or come off from or go into the tissues. So here is a cartoon representation of hemoglobin. The yellow, here's one beta globin, here's another beta globin. Don't worry about the beta and the alpha. Here's an alpha and an alpha. So 2 alphas and 2 betas. In the very center, it looks like Saturn, doesn't it? That little red and blue Saturn group, that represents the heme, the blue does. And in the very center of that heme group, the red circle, the red sphere in the very center, represents the iron, right? Fe is iron. And it's to that little red iron that the oxygen is binding. So each hemoglobin can maximally contain 4 O2s because every hemoglobin only has 4 irons attached at the 4 heme groups. Last thing about red blood cells is that there can be some problems here. And here's a condition that I showed or talked about briefly back when we were discussing a little bit of the genetics and the mutation chapter. And that is that a disease called sickle cell anemia. Now, sickle cell anemia is a genetic condition that affects the red blood cells. And what it's really specifically affecting is the hemoglobin, HB. And the hemoglobin has a mutation. This protein has a mutation in it. There's a 1 nucleotide and 1 amino acid switch in the defective hemoglobin. And what that defect does is it causes the heme group to not bind oxygen very well. So it doesn't bind oxygen very well, so these individuals are anemic, right? They don't have the energy levels because the oxygen can't bind to the hemoglobin as well. The other thing that this weird mutation does is it causes the hemoglobins, normally the hemoglobins are in here, right? In the red blood cell. There's 280 million of them spread inside the red blood cell. But this mutated hemoglobin causes, under stress, the hemoglobin molecules to polymerize, that is to sort of create a chain with each other. And that chain, they all stick together, and remember that red blood cells are kind of flexible. I told you it's kind of claustrophobic when they go through a capillary. They have the ability to kind of stretch a little bit. You already saw in the lab that red blood cells can swell up or crenate down, so you know they're kind of flexible. And so that hemoglobin is going to cause them to stretch and change their shape into this sickle arrangement. I've already told you as well that a red blood cell is 7.5 microns across, and that small vessels are only 8 to 10. When these red blood cells sickle, when they stretch out, they now are too big for the blood vessels, and they start clogging up the small blood vessels. So when these individuals have a sickle moment or these sickling episodes, their red blood cells start to change shape. That's going to block now the flow of blood to their tissues. Now, if that flow of blood is restricting the flow to, let's say, the pinky, then we're going to be okay. But if that flow of blood, if that sickling is stopping blood flow to the brain, the kidney, the heart, the liver, some major organ, then it could be fatal, and certainly could be causing tissue damage. And what happens is that over time, the more of these episodes that a person has, the more tissue damage that they sustain. They're very painful episodes because when you don't have oxygen going to an organ, it's painful. Your body sends off a pain signal. So these individuals go into the emergency room. They get transfusions. They get blood thinners to try to get the blood clots, not clots, but they try to get the vessels to open up. They give them transfusions to try to cleanse out these sickled cells. Once they sickle, they don't go back. So once they deform themselves, they're stuck, and they've got to get rid of them out of the body. And what causes this is, again, it's a genetic mutation, but what exasperates it is stress. So these individuals are told to be stress less, if you can kind of cool it. Physical, emotional stress can trigger it. The other thing that triggers it is high altitude. So these individuals are told, don't go to the mountains. Don't go into high altitude places. Avoid airplanes. Because even though the cabin is pressurized, there are pressure changes, and that can exasperate the sickle episodes. So it's a rather significant disease. One of the things we've learned is that, people who have sickle cell anemia are largely from what part of the world? It's a much more prevalent disease in the 1040 window and along the equator, right? Where typically the African populations, and also there's also forms of this going around Asia in a different form of it, but a different type of disease, but pretty much around the equator. And now we know that people who are carriers of this disease are also resistant to malaria. So there seems to be an advantage to not having the disease, but being a carrier of it. So if you're a carrier of sickle cell anemia, if you carry one of the copies of the gene, you're actually resistant to malaria. Now, malaria is a mosquito, parasitic disease that infects red blood cells. And it turns out that the red blood cells that have this defect are resistant to infection by mosquitoes. So there seems to be an advantage to maintaining this mutation in the population. The problem is when two people who are carriers of this disease marry, 1/4 of their kids will have full-blown sickle cell disease. And so that's the trade-off, if you will, for having malaria resistance from a genetic standpoint. Okay, that's the last little thing about red blood cells is just a reminder, where are they made again? Bone marrow, right? That looks like, what bone does that look like? Looks like the humerus, right? We got that. So remember in the proximal head of the humerus, the proximal head of the femur, you still have active hematopoiesis, you still have active blood production. So for example, you have red blood cells being made in the head of your humerus. They're going to travel through your blood for 120 days. After 120 days, they're going to start looking like worn out dollar bills, and the spleen and the liver are checking for those worn out red blood cells and will break them down by macrophages and start breaking down those red blood cells, but the body recycles it. So we don't want to lose all that iron, right? All that iron that we have invested in all the hemoglobin, we want to recycle that. So the iron gets reused. The heme part gets converted to bilirubin and eventually ends up in your liver and then gives you color for your feces. So the brownish black color of your feces is from the breakdown of this heme group. And then the cellular components are broken down. But basically, we're able to recycle quite a bit of it. It's a pretty cool, efficient system of creation and breakdown, and about 120 days, again, about 4 months. The last thing I want to discuss about red blood cells, and this comes back to the lab objective of blood typing. When we discuss antibodies, right, we talked about antibodies binding to antigens, and we mentioned the 8 different blood types. Those 8 blood types are based upon what? They're based upon a protein found on the surface of your red blood cells. So your blood type, your A positive, A negative, B positive, that is based upon the genetics, again, of your red blood cells. And so on the surface of these cells, you have your antigens. Again, the A and the B and the RH are considered antigens, right? Antigens. Now, the way I think about it when it comes to blood typing is that these antigens were genetically determined, weren't they? You inherited your blood type. So remember that your antigens are genetically determined. In this case, you inherited them from your parents, and your body is going to make antibodies against what? Is it going to make antibodies against your own antigens? No, your body's only going to make, normally, is only going to make antibodies against foreign antigens. So basically, your immune system, when you're born, you don't have much of an immune system. And that's why we know that there's advantages to breastfeeding, because the mother's antibodies are passed through the breast milk to the baby and help to establish an early immune system in the baby. And then the baby's immune system continues to make antibodies through the early years. So our immune system starts off rather weak, right? Kids are more prone to getting ill. Our immune system is more robust and ramps up throughout our life. Hopefully stays really great for a long, long time. And then as we age, also our immune system starts to decline a little bit, and we're more prone to illness again. But these antibodies are, again, going to look for and be produced against foreign antigens. When it comes to blood typing, there were 3 antigens, right? The A, the B, and the RH. And those are the 3 that we're worried about, right? And it depends upon which combination of those 3 that you've inherited gives you one of the 8 blood types. Either you have it or you don't. And there's 3 different options. So you either have the A or you have the B. So there's 2 different options, and there's 3 of these different choices. So that gives you the 8 different possible blood types. Now, normally, antibodies would not be produced against your own antigens. It happens, right? People have autoimmune diseases where their body has, for whatever reason, been tricked or is troubled and is creating antibodies against self. And that's what happens in rheumatoid arthritis, in lupus, and any number of diseases where the body is actually fighting itself. But normally your body only makes antibodies against foreign things. And so the problem is if you receive the wrong blood type, if you get a transfusion with the wrong blood type, then your body will fight off those foreign red blood cells. And what the immune system does is it clumps. The antibody will bind to the foreign antigen. It'll bind to the foreign red blood cell and form a clump. That clumping reaction is important because it tells your other immune cells to go and destroy. So when antibody binds to antigen, it sets up a signal that tells your macrophages and your other cells of your immune system to come in and gobble up and destroy that foreign thing. The problem is, with blood, you're receiving so many cells. When you get the wrong blood transfusion, you just didn't inhale a little allergen or get a little bit of something, you got this massive number of red blood cells given to you of the wrong type. And those antibodies now will start clumping with those foreign red blood cells and can cause clumping agglutination reactions, which sort of like the sickle cell anemia can block your blood vessels. And if you've been given enough of the wrong blood type and your own immune system is robust enough to have a lot of antibodies against it, then you can have significant flow problems to critical organs. And if you've been given enough of the wrong blood and those agglutinations block up the brain, kidney, heart, lung, then you can have some serious damage to your organs, or worse yet, even die from this. Now, eventually, those clumps would be destroyed. But in the meanwhile, you haven't had oxygen to your tissues. So you saw this in lab, and I hope this is making sense for you. And if you'd like me to, I'd be glad to go through another blood typing scenario with you. But in this scenario, in this chart, what am I showing you? If you inherited the A antigen, it's shown here as little blue dots on the surface of your red blood cells, then you would be classified as having type A blood, and your body would consider B blood foreign and would have made antibodies against B. If instead you inherited from your parents the blue, or the little purple spikes, the B antigen in this cartoon, then you would be considered a person with B blood, and you would make anti-A, antibodies against A. If instead you inherited both the A and the B, that's fine. They're both on the surface. You have AB blood. Because you have both, your body would not make antibodies against either one. If instead you inherited neither A or B, you have type O. Again, think of O as being not like an O antigen. There is no O antigen. But think of it as a null, right? As a big zero. There is no antigen, A or B. And as a result, you would make antibodies against both A and B. Superimpose on top of these four options either the presence of the Rh factor, in which case we say that you're positive. So a positive blood type, positive means you have the Rh factor. If you don't have the Rh factor, then you are one of the negative blood types. So again, positive only means you have the Rh, negative means you don't have the Rh. As we discussed, O negative is a universal donor. Naked little red blood cells. There are no antigens on the surface, so you can give O negative blood to any individual. And it's not going to alarm their immune system that, hey, you just gave me something foreign. AB positive, these individuals' red blood cells have all three antigens, the A, the B, and the Rh already present. Their body says, hey, I'm cool with it. I can take anyone's blood type. And so they are considered the universal acceptor. Should I go through an example? Or do we have this? What do you think?
Let's see one.
Shall we do one?
Yeah.
Okay, let's do it. So I'll go to my chalkboard here. Let me get some white chalk out. There we go. So, the ones I did in lab, let's do one I didn't do in lab. So, and I always do the same thing. I go through the same procedure every single time. So I draw my little red blood cell. Let's do AB negative. I don't think I did that in any of the lab groups. So let's do AB negative. So same questions. What antigens would be present on the surface if you had AB negative blood? A and B, right? So you're going to have the A and the B. What do you not have? Rh. So your body would make antibodies against Rh. Because it's making antibodies against Rh, it would not be able to receive blood types that had what in it? Rh. That is all the positives. So you would not be able to receive A positive, B positive, O positive, or AB positive. Right? We could not receive those because they all have positive in them. They all have the Rh factor. If those were to be brought into the body, the blood would clot. You would be able to bring in the other blood types, and they would be what? You would be able to bring in all the negatives. So you could bring in A negative, B negative, AB negative, and O negative. What would I not, okay, so that's what I cannot receive and what I can receive. And then when I go to give this blood away, what am I giving away? A and B. So I can only give it to individuals whose immune system are already trained, right, to be okay with A and B. Which blood types have both A and B? Right, both of the ABs are okay, negative and positive. Remember that over on my fourth question, you're always going to have O negative, right? O negative will always be because it's the universal donor. And in all of the examples, you'll have AB positive over here because it's the universal acceptor. Are we having any problems with blood typing? I'm going to find out next week on the lab quiz. And I'm hoping not, right? So I think I went through 4 in the lab. This is the 5th one that I've gone over with you. If it's not making sense, please say something. Please let me know. I'll be glad to go through it. I'll stand on my head. Well, that wouldn't be good. But I'll try to say it a different way. Okay? And you still have 3 other ones that you could fill in. So I don't think we've done, I don't think we did A negative. I don't think we did B positive. And depending on what lab you're in, I either didn't do O negative or I didn't do AB positive. So you've got other ones to look at to practice this. Is it coming into place? If you'll just ask those same 5 questions and logically progress through it, I don't think you'll have trouble. People who seem to have trouble with this are the ones who have memorized it or think they have memorized it. And they think they've memorized these patterns. And while that's a legitimate way, I suppose, I don't see problems with people who have logically working through it. I only see problems with people who have memorized it. And then they forget, right? It gets to be too many crazy little A's and B's and O's and plus and minus signs, and they go crazy. What do we think? Are we okay? Nancy, you got it?
Mm-hmm.
Those who
I
have,
just wanted it on the PowerPoint.
that's,
so I-
and that's a really good thing to, yeah, it's very good. I'm glad I did that. It was even recording.
Oh, good. Even better.
Even better. Now, next week you'll be doing blood typing in the lab. You'll have the option of doing that. And the way we're going to test your blood is whether or not it has the antigens. Right? So we're going to take drops of your blood. Your blood already has the antigens on it. We're going to go down to Walmart and we're going to buy some antibodies. And we're going to have an antibody against A, an antibody against B, and an antibody against Rh. And what you're going to do is basically next week you're going to get a little card and you're going to put your blood on that card. Let me do it this way. You're going to get a card and you're going to put your blood, I'm going to go color here, a little bit of blood here, here, and here. Your blood already has the antigen on it. You're then going to add to it, I forget the colors, but one of the antibodies is yellow. One of the antibodies is colored as blue. If I can get this to work for me. Oh, that's gray. Okay, different color. And one of the antibodies is, I don't know, let's go with a different color if I can get it to work. Okay, kind of a pinkish color. And you're going to rub these together. And if the antibody sees the antigen, what's going to happen? Clump. And so what you're going to be looking for next week is the presence of this agglutination, this clumping reaction. If you saw clumps, when you mixed these things together, when you mix the antibody and the antigen together, if it clumps, you know that you have the antigen on your blood. So if you add a antibody and it clumps, it means that you have the A antigen. If you add the B antibody and it doesn't clump, it means you don't have the B antigen. And we're going to look for the combination of clumping reactions to determine your blood type. So you'll see all that next week, and that will become, I hope, very, very crystal clear. You saw this slide already as well, and that is what goes wrong, as I've already described. This guy has type B blood, and somebody didn't do a good job of reading the chart, and so this guy has B blood, but someone is giving him A donation, a blood type donation. And those little A red blood cells are shown in purple just so we can keep track of them. The little yellow slingshots in his blood represent the antibodies. And what you see is that his antibodies will very quickly seek out and find the foreign antigen on the foreign type A blood and will cause this agglutination reaction. And what's happening right now is that whatever is down here is not getting oxygen. So that tissue is now becoming oxygen deprived. And again, the number of and the location of these blockages can have a very significant effect on the individual. So you don't want to give anybody the wrong blood type. Okay? So that finishes up everything that I want you to know about red blood cells, from hematocrit to blood typing. Anything else about red blood cells? I can give you a little break right now, and then we'll jump into white blood cells and platelets, and we'll finish up this chapter. So let's continue. We finished up the conversation on red blood cells. Now let's move over to the white blood cells. And honestly, there's less here for you to learn this semester. We'll save more of this story for next semester. So the white blood cells, turns out that these cells are they're true cells. They have a nucleus. They have a metabolism. They have DNA. They can divide. They can replace themselves. So they are the only true cell in the blood. They're going to be by size about 1.5 times up to 3 times bigger than red blood cells. So when you see them on a smear of blood, they'll be bigger. Now, these cells do not contain hemoglobin. They have nothing to do with transporting oxygen or CO2. Their numbers, again, are far more rare. And if you had a reduced number of white blood cells, you might hear that you have leukopenia. Okay, leukopenia. Leuco, white, penia, a deficiency in. So leukopenia. This would be anything under that 5,000 per microliter. It could be a form of leukemia. There's all sorts of reasons why you might have a low number. You might more often, though, hear about someone having leukocytosis. Leukocytosis would be an increased number of white blood cells. And the reason you would have an increased number of white blood cells is your body's fighting off an infection. So a bacterium has invaded your body, your immune system is mounting its defense against it, it's creating a massive number of white blood cells. And so that we'll think of that as being the infection being the most obvious reason for that. Let me go back, though. Now that I'm using these words, I don't think I used the word back on the red blood cells to describe a deficiency. So I'll just put it here on this slide. If you have a low number of red blood cells or a low hematocrit, then you would have what? You would say to have anemia. So, right, we've heard that term. And if you are anemic, they're going to tell you to go eat some liver or some spinach, right? Because both of those have a lot of what? Iron. Again, if you can raise your iron levels, then you can make more hemoglobin, carry more oxygen, get your energy levels back. Okay. I mentioned, too, that men have a higher hematocrit, and the reason is men have bigger brains. By their anatomical size, they need more oxygen to run those bigger brains. They have more muscle mass on average. Testosterone also upregulates red blood cells. So the more testosterone one has, the higher their hematocrit. And this is one of the reasons why we hear all these low T commercials now on TV. You know, a guy with lower testosterone levels wouldn't have as much energy because testosterone is linked to having a higher hematocrit. Okay, so I just wanted to use that word anemia there, since over here I'm describing a term when there's a decrease in the white blood cells, leukopenia. Now, there are 5 different types, 5 different classes, if you will, or groups of white blood cells. And we're going to break them into two, what I'm calling here, classes. So 5 different types of white blood cells broken into 2 classes, granulocytes and the agranulocytes. Pretty easy. We've seen that word grain or gran before, meaning particle-like shape. And we saw the stratum granulosum in the skin, and it was that layer where there was little granular-looking appearances. So the granulocytes are cells that are going to have a granular appearance. They're going to have a grainy-looking appearance, whereas the agranulocytes would be lacking that. So let's look at this. In this one picture, here are the 5 different types of white blood cells. Your job this semester is not to recognize them. So I'm not going to ask you which of these is which. Okay, that's for next semester. But I do want you to break this into the 2 groups. So, get some color on this. So the 3 on the top are the granulocytes. And if you look at these cells, they appear to have a granular appearance to them. Now, the granular appearances have different colors, and we won't worry about that right now. But the neutrophils, the eosinophils, and the basophils are the 3 white blood cell groups that have granules. So we're going to say that those are the 3 granulocytes. The way I remember this, turn those letters around, and you have Ben. So Ben has bumps, Ben has acne, Ben has granules, whatever you want to think about it, right? And if you know somebody with Ben and you love them and you don't want to think about them, think about Neb, okay? But somebody, right, Neb or Ben, has these granular appearances. The 2 on the bottom are the agranulocytes. They don't have the bumps. You don't see the presence of those bumps in the lymphocytes or in the monocytes. So we will talk a little bit about each of these cells. We'll have a little bit of head knowledge about them, but I'm not going to ask you to recognize them visually. Let me start off with a couple of things about the granulocytes. The first one is the neutrophil. Easy to remember. All I want you to know is what's here on this slide. So the neutrophils are the most numerous, right? Neutrophil numerous. They're by far the most numerous of the white blood cells. They are rather neutral in color, and that's how they got their name. I just told you in the vocab that phil means what? An affinity for or a love of. These cells, when scientists about 100 years ago were playing around with microscopes and looking at cells under the microscope, they realized that they had to add some dyes, some stains, to see cells better. And they came across a combination of stains that allow one to see blood cells really, really well. When you look at white blood cells, they're going to have different colors because of the dyes that have been added, not because they're floating around in your body as purple cells. And it turns out that the neutrophils are rather neutral in their affinity for the dye. So they have the little granules, but they don't have much of a color. They're neutral in their color. Okay? The eosinophils got their name because one of the dyes that is used to see blood is eosin, and eosin is a reddish, orangey, pinkish colored dye. And it turns out that the eosinophil has an affinity for this dye, leaving them kind of pinkish colored. Okay? So these cells got their names based upon the dye to which they have an affinity. So eosinophils have an affinity for this dye called eosin. Eosin is a reddish, pinkish, orangey colored dye. Then the third one are the basophils. And what I want you to remember here is that the basophils are the least numerous, so their numbers are in the basement, right? The basophils, their numbers are in the basement. They're the least numerous of the granulocytes, of all the white blood cells. Now, what's interesting about basophils, the little side that I want you to know, is that they carry and release histamines. When do you deal with histamines? When you're having an allergy attack, right? And then what do you run to Walgreens to do? Buy some antihistamines to counter the effects. So what do histamines cause your body to do? Weepy eyes, right? Draining nose, what is your body trying to do? When that allergen comes into your body, the basophils start releasing histamine that causes your body to do what? Try to get rid of it. But then what do? We go to Walgreens, we buy antihistamines, and try to hold on to it. It doesn't make much sense in a way, right? So what we're trying to do is cover up the symptoms, but honestly, the best thing we might want to do is just let the body do its job and get rid of this thing that's causing the allergic response. The problem is we want to be somewhat social and not be, slobbering all over everybody. It's not very much fun to have the runny eyes and the runny nose, but the body is desperately trying to get rid of whatever it is that has entered the body, and we try to cover it up with antihistamines. Oops. So now so that's all I really want you to know about the 3 different granulocytes. Okay? 3 different granulocytes. The other 2 are the agranulocytes, and this is going to include the lymphocytes, and the monocytes. They don't have really large granules that are easily seen under the microscope. So lymphocytes, you've heard of these. You've heard of lymphocytes maybe with different names. Somewhere along the way, I know you've heard about T cells and B cells. You don't know much about them perhaps, but you may know that people with HIV, their T cell count starts to drop, and when it becomes critically low, they have full-blown AIDS. And the T cells must therefore be an important part of the immune system. And what the HIV does is that it destroys the body's ability to fight off infection by destroying these T cells. So that's a type of lymphocyte. Then there are also B cells, and we're not learning a lot about these things, but B cells are the very cells that make your antibodies. So when we're talking about antibodies being produced by your body as part of your immune system, those antibodies are specifically made by these B cells. And then there's a whole bunch of these. There's other ones, really cool ones called the natural killer cells. We won't go into that this semester, but if you take microbiology, you'll get a very robust chapter on how the body fights off infection. And you will learn a lot about T cells and B cells and the immune system in that class. Finally, we have the monocytes. They are easy to remember because they are monstrous in their size. They're the biggest in size of all of the white blood cells. So I just remember they're monstrous in size. Don't worry about their abundance in the body, just know that they're really, really large. And these monocytes are or become macrophages. And macrophages are cells that do what? They're large cells that eat. So macrophages are like little Pac-Man. Now you guys are all way too young to know who Pac-Man is. You know who Pac-Man is? Oh, I thought that was just for the old folks. Right? Pac-Man was all the rage. I mean, that was video game world, right? But way back in the 80s, right? Pac-Man and Pong. And that was our life. But these macrophages go around and eat up old stuff, decaying stuff. Okay. That's it that I want you to know for white blood cells. Now, I do want to say this to you. Why are these things called white blood cells? Because they're what? They're called white cells because they're clear? They're white. Okay, but if I look at any of these pictures back here, none of these cells in this picture with all 5 of them right? When I look at this slide with my 5 slides when I get back there, those cells don't look white to me, right? In the background, what do we have? All these little guys are these are all the red blood cells, right? And then they're very, very abundant. And then we see these larger cells. These are all the different kinds of white blood cells. And they have these different colors because of the dyes that have been added to them, not because they're naturally purple or red or orange. So why are they called the white blood cells? Red blood cells have hemoglobin, so they're red. That's why they're the red blood cells. But why are these the white blood cells? When you have an infection, you get a lot of neutrophils coming to that part of the body, and that can produce pus. And pus is white. So pus is actually white blood cells coming to the rescue, creating all of that cellular debris. So all that crap that's being formed in the pus is largely the white blood cells. So that's where they get their name, right? Red blood cells carry hemoglobin, they're red. White blood cells, part of the pus, part of the antibiotic or the immune response to infection. Okay, now that leaves us just with platelets to describe. And platelets, again, a totally different formed element with a totally different purpose in its life, and its job is to be involved with blood clotting. By size, they're much smaller, about 1/4 or less the size of a red blood cell, so down in the 1 to 2 micron area. Now, there's another name for platelets. You'll see them called thrombocytes. Not a great word, because they're really not cells, but still, they have this name, thrombo. Now, thrombo we haven't gotten to yet in our vocab, but thrombo means clot. So the cells that make clots, right? Thrombocytes. A thrombosis would be a blood clot. Now, where do these platelets come from? They, like red cells and white cells, are made in the bone marrow. But they're produced by a cell called a megakaryocyte. So you tell me right now, what must a megakaryocyte look like? Or what must it have? Mega, large, karyo, nucleus. So this is a cell with a monstrous shaped nucleus, and so that's where it gets its name. These cells are only found in the bone marrow, and your body is constantly making these platelets from the megakaryocytes. Again, they're not really cells. They're little fragments of the megakaryocytes. The platelets will be activated when the blood vessel becomes damaged, and that's when fibrinogen will be cleaved to fibrin. So it turns out that you've got the platelets and you've got the fibrinogen coming together, if you will, to get trapped together to make the blood clot. Now, a blood clot is different than an agglutination, but they both stop the flow of blood, don't they? So agglutination, antibody, antigen coming together can stop blood. Sickle cell anemia can stop blood because of the shape of the cells, and now we have another way of stopping blood, and that is through a true blood clot. Now, the number of platelets, do know this number in relationship, about 120 to 300,000 platelets per microliter of blood. Again, that's in comparison to 5 to 10,000 white blood cells, and in comparison to the 5 to 6 million red blood cells that would be present in the same volume. They float around for about 8 to 10 days, and if you had a low number of platelets, you would have thrombocytopenia. Thrombocytopenia, right? Thrombocyte, right? The bad word for platelets, but it is used, and penia meaning a deficiency. So thrombocytopenia, a lower number of platelets, and what would your symptoms be? If you have low platelets, you wouldn't be able to heal, you wouldn't be able to clot well. So you would have an increased bleeding. If you were to cut yourself, you would keep bleeding. It's not the same as hemophilia. Hemophilia is a different whole condition with genetic issues, but thrombocytopenia, you would have a difficult time clotting your blood. Also, a person with thrombocytopenia would bruise easily. You know, you look at some older folks and they bruise very, very easily. They knock their hand against something and they get big bruises. We're always bruising, right? We're always knocking our hand on something and our body clots internally very, very quickly. So we don't see any evidence of blood pooling under our skin. But a person who has any kind of clotting problems or has thrombocytopenia, every time they bang their hand up against something and they get a micro bleed, it's not healing, it's not clotting off properly, so that blood is seeping out into the tissues. And if you ever had a bruise, it's the same thing, right? That blood underneath, you get a bruise, it turns beautiful colors, right? Yellow, black, green, purple. All those colors from a bruise are the blood underneath the skin going through layers of or times of degradation. And so you see lots of bruising from a person who has thrombocytopenia because their blood is not adequately clotting under the skin. This is an image of a megakaryocyte. Again, big old cell, right? Big old cell, big old nucleus. And what's actually happening is that this megakaryocyte is blebbing off these little tiny pieces. And all these little blebs are platelets. So you basically have a megakaryocyte kind of blebbing off these little fragments of itself. And those little platelets are now going to go into the circulation, and they're going to hang around for about 8 to 10 days and then get destroyed. Constantly being made. If you were to take a smear of your blood and look at blood under a prepared slide, again, this image over here has been stained. For you to see this blood and for you to see those white blood cells as being purple, you know that the stains have been added, the dyes have been added to see the blood cells optimally. The vast majority of what you're going to see, so take a drop of your blood, stick it on a slide, smear it with a cover slip, and what they're not showing here is the stain, right? It doesn't say that. It says it, but it doesn't show it. They've actually added some stain for contrast. Put the cover slip on top, look at it under the microscope, and the vast majority of the cells you're going to see are red blood cells. You're going to see larger, darker staining cells. Those are some of the white blood cells. And in this picture, probably even too small to see, but I'll draw a couple in, little dots, and those would represent some platelets. So let's put all this together. When it comes to the white blood cells, again, we have the granulocytes, the neutrophils, the most numerous, the eosinophils, didn't talk about them in any particular way, and basophils have the lowest number. Their numbers are in the basement, and those are the cells that create the histamine. Under the A granulocytes, there are the lymphocytes. These are your T and B cells that are directly attacking viruses and bacteria and also make antibodies. And then the monocytes, which I told you were monstrous in their size and became macrophages. So those are really just the keys that I just described to you. That's all I want you to know as well. Keep knocking myself out of this. This is a close-up micrograph of a blood clot. And what you're able to see, this is a scanning electron microscope. You would never get down this close and see these cells this large. But what you're looking at here, all these strings, all these fibers are fibrin, not fibrinogen. Remember, fibrinogen was soluble. Fibrinogen is not forming a clot. Fibrinogen is floating in your blood all the time. But something has caused blood to clot. There's been damage. The fibrinogen was cleaved to fibrin. Fibrin becomes the network, the fiber, right? It becomes the catch, the net. And into that fibrin clot, you get platelets that get caught up. So all these aggregates of white things are the platelets all over the place. And also in this picture are a large number of erythrocytes. Now, there are some white blood cells in there as well, but I can't pick one out. Okay? They're not anywhere near as common. So that's what a blood clot would look like under the microscope. Great. I mean, just a really nice, complete blockage of the blood flow. Now, after a while, I won't talk about that today, but after a while, the clot is broken down, right? It is dissolved, and then blood flow is restored, but not until the tissues have been fixed. Now, the last couple of words I want to use here, the last couple of slides, I want to describe this overall process of making blood. The overall process of making blood is called hemopoiesis. You will also see equivalent the term hematopoiesis. So either one, same thing. So either hemopoiesis or hematopoiesis. And again, poiesis means what? Formation of, right? So we saw that in our vocab today. So hemopoiesis or hematopoiesis, the making of all of the formed elements. All of hemopoiesis is happening where? In your bone marrow. As an adult, remember that's still happening in the proximal end of your femur, the proximal end of your humerus. It's still happening in your frontal bones and your skull bones. It's still happening in your clavicle. It's still happening in your ribs, your sternum, and your vertebra. Mostly your irregularly shaped bones and your flat bones. And all of your formed elements start from a single cell. Here I have that cell called a hemocytoblast. This would be a blood stem cell. And from this one cell type, all of your formed elements are produced. Hemo, blood, cyto, cell, blast, osteoblast, chondroblast, right? Other things are made from it. So the hemocytoblast is the cell from which all other blood cells are produced. Now, this cell will undergo what we call differentiation. We talked about that way back long ago. Differentiation is the normal process that cells go through as they become different from one another. So there'll be different hormonal signals, different cellular signals that the hemocytoblast will receive in the bone marrow, and it will begin to make different types of blood cells. The first choice that the hemocytoblast has to make is, and I know this is too small to read, is it going to go lymphoid or is it going to go myeloid? Myeloid or lymphoid? Now, so up in the top, this is the myeloid side and this is the lymphoid side on the right. Once it makes that decision, if it chooses to go to the lymphoid side, this is really easy because from the lymphoid side, all you're going to make are the lymphocytes. So down here are the lymphocytes. So lymphoid makes your lymphocytes. Easy enough. You may hear someone having different types of leukemia, and it's described as being a lymphocytic or a myelogenous leukemia, something like that. And what it's telling you is that a different type of blood cell is out of control. So if the hemocytoblast goes toward the lymphoid side, then it's going to make the lymphocytes. If it goes to the myeloid side, it can make everything else. So again, that's not so hard. I always remember myeloid many. So it can make all the other blood type cells. From the myeloid, it can then go on to make red blood cells. It can also differentiate to make your megakaryocytes, which then bleb off to make your platelets. The myeloid cell can also go to make other cells, which eventually become your neutrophils, your basophils, your eosinophils, and your monocytes. Okay, so all of the other blood cells are derived from the myeloid side. That's all I'm going to say. All the stuff in between, all the intermediates, don't worry about any of the stuff in the middle. Just know that we're able to create the end product cells, all from one stem cell. So if someone has leukemia and they need a bone marrow transplant, what are we going to do for them? What's the process in a bone marrow transplant? Anybody know? First, we have to find a donor, right? And finding a donor is not easy. But you donate or you become part of the register, and what they'll do is they'll actually take some of your bone marrow and test it. It's a little bit painful, but now your bone marrow and your type is in a registry somewhere. And then if somewhere in the world needs your exact blood type, it's not just a matter of matching A, B, or B blood type. There's a lot of other matches that go on in matching up your bone marrow. And it's pretty hard to get a perfect match. But if you are the person who needs that bone marrow and you become the donor, and they know there's a match, they're going to take that patient, they've got leukemia, they've got some sort of blood disorder, and they're going to nuke them like you can't believe. And they're going to destroy all of their bone marrow and their blood cell capacity. They're going to wipe out their red cells. These folks are going to be very anemic. They're going to wipe out their white cells. They're incredibly immunocompromised. They're going to wipe out their platelets. They'll bleed if they get hit at all. Bleed out. So they have no white cells, red cells, or platelets. And then we're going to implant into them bone marrow. That bone marrow will go in and hopefully graft and take, and that person now will start producing your cells, basically, right? So now, this is interesting, their white cells have a different DNA. If you've had a bone marrow transplant, you can get away with murder. Right? Your skin cells are still you, right? Your skin cells are you. But if you're leaving behind blood, that blood DNA is going to match the person who gave you the donation. Okay? So it could really mess up a crime scene a little bit, couldn't it? And we're going to bring that person basically to the brink of death and then give them that bone marrow transplant. It'll graft, it'll start then producing their red cells, energy, white cells, immune system, and platelets, their ability to clot blood. The overall process of making all blood is called hemo or hematopoiesis. The formation of just making red blood cells, we would use this term, erythropoiesis, right? So just the formation of your red cells, 3 million per second. Crazy numbers. Again, this is coming from the myeloid side of your blood production, and it takes about 5 days. So this also tells you that when you fly out to Denver, right, it's going to take up to about 5 days before your red cell count is going to regenerate itself, so you're not so loss of energy. If we're talking about just the formation of platelets, we're going to use the word thrombopoiesis. If we're talking about just the making of white blood cells, we're going to use the word leukopoiesis. Okay, so poiesis, just a generic term for the production of. And that brings us to the end of the blood chapter. I feel pretty good about that. What do you think? I really don't, I don't feel a need to spend the next 5 minutes talking about the heart. But on Monday, I'll present the chapter on the heart. Again, a lot of it you've heard. We've already looked at the heart. I know we went through quickly. In lab, we kind of did a real quick lab this week. We didn't spend as much time. You should now, hopefully next week on the lab quiz, have no trouble with blood typing. We've talked about it. We've talked about the blood. You definitely want to go back and look at the heart structures, the flow of blood through the heart, and we'll talk about that in lecture on Monday. And then next Wednesday, we'll talk about vessels, naming some blood vessels, how are arteries and veins different from each other. And that's going to put us in a really comfortable place to be ready for the exam on the 21st. Questions for me? This weekend, really no exam hanging over your head, but you do have, of course, your short paper and your take-home exam, so I guess you do have that to do. And don't forget, there are the 3 mastering assignments. You certainly can now go and do the blood mastering assignment. I wouldn't do the other ones yet. And you can also start looking at that exam, that quiz. There's no reason. You can go in there and look at the blood questions and try them. And then you can come and go. And then make sure you submit that before Sunday noon on the 20th. Okay.
What was the third mastering assignment? 2 more homework?
There's 3 homework. There's one in the blood, one in the heart, one in the vessels.
I only saw 2.
17, 18, 19.
Oh, okay.
Should be 17, 18, and
I
19.
only saw 17 and 18.
Yeah, on the syllabus, it only says 7.
Syllabus is off on numbers. Now, you're saying mastering doesn't show it.
Yeah, I don't see it on mastering either.
I put it there this morning. I'm pretty sure it's there, but maybe you looked when I was putting it together. I put those on there this morning around 10:30.
Yeah, that's a problem.
So maybe you saw it before I got the third one on there. And then the syllabus is incorrect. I think it says 17, 18, and it's really 17, 18, 19. Okay? Okay, very good, folks. Have a wonderful weekend.